Hey everyone, it's Mr. Fabeck here, and this is Fabiology DNA Replication. So we're going to study life in this one. We're going to look at what makes up life on a microscopic level. So by the end of the video, you're going to be able to understand or explain the purpose of DNA replication. So why do we do it? And then describe how DNA is replicated in terms of the enzymes being used and how the bases pair up. So you'll know you mastered this if you're able to draw and label a picture of DNA being replicated, or just, if I give you a random strand of DNA, can you copy it to form the complementary strand? So here's the why. What's the whole purpose of doing this? Well, we have three things. One is mitosis. So mitosis is all about growth, repair, and replace. So as we are growing, we're making new cells. We're essentially copying the exact same blueprint of DNA. So it kind of looks like this. It's kind of rad. We copy our chromosomes and then we, boom, split apart to make an exact replica of the cell. So anytime you get a cut, a broken bone, or you're just growing, because we're obviously not the same size we were as a baby, we had to do mitosis and we had to copy our DNA to do that. The other idea is meiosis. Meiosis is when we're making sperm and egg cells. So we're trying to make the ingredients to make a baby, if you will. But we only give half of our DNA. So we first copy it, and then we split it in half so that one sperm cell has half of your DNA, and the egg cell has the other half, so you can make one complete set of DNA. And then the last one is why we love all those crazy cool crime dramas like Bones or CSI, or one of my favorites, Dexter. Oh, love it. But it's all about copying DNA for research, for more samples. And think about it. If you only have one little spatter of blood, Dexter's a blood analysis person, or one hair, or one sperm cell, or small sample of DNA, you want to be able to copy it so that you can run a multiple test so that you can really trace down and narrow down suspects. So, and especially as time goes on, maybe you want to store it so that we can figure out who did it later at time. So those are the big three of reasons why we want to do DNA. So let's get right into it. What does it look like? Basic structure is what we call a nucleotide, which is three parts: a sugar, a base, and what we call a phosphate group. I like to say it looks like a house with a balloon and a mailbox. That's how I remember. It's like the up house, if you will. And so these nucleotides are all strung together, and when you put them together, they look like this. The base pair is hooked together with really small or weak bonds. It's just hydrogen. Hydrogen bonds are super weak so that you can pull them apart, which is great because if we think back to the purpose, we want to be able to copy our DNA regularly to grow and repair and replace tissue. So these weak bonds are holding the bases together, but as we hook them together, because of the chemistry, you can see one side gets flipped and then it starts to actually twist. So we call it a double helix, and that's what Watson and Crick are credited with discovering, although Rosalind Franklin was the one that gave us that X picture there to show us that there was this twist happening, and that was kind of the final piece to what the double helix was all about. And once we were able to get this structure, then we were able to ask, okay, well now how does it change over time, and how do we replicate it during mitosis and meiosis? So thinking about replication, there's a couple possible ways that this could happen. So when scientists were thinking about this, they thought, okay, if we have an original DNA strand, we might say it's called dispersive. What if we have our original and then it's just randomly new parts? So it's all kind of a little bit of old and a little bit of new. Well, that's not exactly what it turned out to be. And then there's the other one of conservative. Okay, so what if we just had all the old together and then all the new together? Well, again, it's the same idea. Well, would that take more energy to do that? Is it organized it that way? So conservative and dispersive didn't really match up with what we were seeing. And what we ultimately found is what it's called semi-conservative or partially conservative, where one strand is completely old and you use that as a template to make a copy of the other strand, which is brand new. So we get one old and one new. And so when we found that out, we learned that every one parent or double helix parent makes two daughter double helixes. So we go from one X, right, one twist, to two, right? We have two separate ones going on here. And that's what we call that replication. We have that one strand and boop, now we have the other strand and it's all twisted up and we're all set to go for whatever we're making. So again, we're trying to figure out how do we replicate. So let's look at that second bullet point there. How are we doing this? We have enzymes. The whole job of enzymes is they have one specific job. That's it. So we have 3 specific enzymes that we need to talk about. And so we have helicase, DNA polymerase, and ligase. Helicase, I like to say, is the zipper, okay? It is the one enzyme that's going to unzip the helix, right? If the helix is the twist, helicase unzips that helix. So you can see in here we have our little helicase, and as it's trucking up and it's breaking apart those hydrogen bonds, we're getting what we call a replication fork, but we're also getting that split happening. And this opens it up so that we can use another enzyme to start putting in the code. So we have the zippers. We have first unzip. Then we need our writer. We need the pencil, okay? So we need the writer, or DNA polymerase, to make the polymer. That's why it's polymerase, right? It's making the DNA polymer. So we can see it's uncoiling. Helicase is uncoiling it, and then our polymerase is coming in, and it's always adding nucleotides to the newly made DNA strand. So it's just pecking in there, A, T, G, C, A, T, T, and it's just going down the line. It's going so fast that it sometimes makes mistakes, and that's what we call mutations, but we'll get into that in another unit. But ultimately, it's very specific in how it does it, and it goes from 5' to 3'. And what that means is we have what are called Okazaki fragments, because remember back when we have the replication fork and it's going and it's splitting apart, well, because our DNA is going, the balloons are going up in one side and balloons are going down in the other side, it really throws things off in how we replicate it. So we have what are called Okazaki fragments, or small fragments on our DNA strands that are incomplete, because as it's opening up, if it only adds the 5' in and only can go in one direction, and they're in opposite directions to start, well, that throws things off. So normally it would go like that, and as it's untwisting, the 5' end is plugging in the code. But the other way, we have to wait, right? So we had, once it opens up, right, we can start copying it, but now we missed a gap, okay? So now we have to go back and add that 5' in. And we go back using what's called ligase, or the surgeon. So when we look back at those Okazaki fragments, DNA ligase is going to connect those strands so that we can fill in the gaps and seal it all up. So when we look at a whole picture of what's happening, you can follow up here. This red strand is the new strand, okay? It was adding the 5' in, and it's going along, and it's just zipping it up and writing the code in, and this polymerase is going to keep on going all the way down. But now you can see visually now we started to unzip it, because this discontinuous strand, it'll start to go, but then another spot opened up, and it would have to go back, and polymerase has to keep going back to go back in, type in the code, and then our ligase finally seal together those parts so that we have a full set of DNA. And I want you to notice we have what's called a leading strand, and it's called leading because it's going to win the race, because as this is opening, it's copying and it's running right with it. But the lagging strand, it's as if it's opening, it's like, oh crap, I got to go back and do something, and it has to go back and fill in the spots that we're missing, and then we have to have a friend help us completely complete it up. So this strand is lagging because it's actually copied slower than the older strand. So putting it all together, so if I gave you a code like this, can you copy it, right? So remember, when we think about our base pairs, we're always going to bind the opposite. So we're going to bind, in this case, Chargaff's rules are A to T, or adenine to thymine, and G to C, guanine to cytosine. A to T, G to C, all the time, okay? And that's because of their chemistry. And notice that the ends are opposite, okay? Again, that's because of the way they get set up with those balloons. One goes one way, one goes the other, because of their chemical structure. So here we go. These are just some practice problems. I give you a 3' end and a 5' end. Can you copy the rest? And don't forget to add those. Remember, you're copying 5' to 3'. And the same thing with the other strand, completely different. Can you plug and chug and be that DNA polymerase and put the right code in there? Okay, so you will be the enzymes. So thank you very much, and good luck to you.